Terry and I, he took me on my first field school, I have to say, in 1984. So we've known each other a very long time, and recently kind of came back together as colleagues on this topic of evidence of Polynesian contact with the Americas. So I'm going to take you guys on a fairly rapid walk through a little bit of history of the project and what we've been finding out, and in a sense where things are going. And some of you may have heard of a paper that came out electronically last week, officially 2 days ago, on DNA, ancient DNA analyses from Rapa Nui and Easter Island, and the implications in the press coverage of that. They've been discussing implications for the Americas, so I'll talk about a little bit about that at the end of the talk. So here we go. And I'd just like to thank Terry and thank everybody here who invited me and have made this talk possible, and all of you who are here to listen. So as I say, here we go. I'm actually a biological anthropologist who my main area of research is settlement of the Pacific. And I've spent the last 25 years or so trying to better understand the settlement of the Pacific using biological tools. And I just happened to be in the right place at the right time when PCR and mitochondrial DNA and ancient DNA and all of these technological developments made this possible to address this from a biological or genetic perspective. So I've been there from the beginning and have some wonderful graduate students now who kind of drag me along as the technology has changed so rapidly, but I'll be talking a bit about some of that data. So as I say, my main interest is in the settlement of the Pacific, and for those of you who aren't familiar, we have divided the Pacific region into two different areas. The Polynesian Triangle, most people are familiar with apices in Hawaii, Easter Island, Rapa Nui, and Aotearoa, New Zealand, which was really the last major region of human expansion. The settlement of New Zealand, the last landmass that was settled by humans, occurred only about 750 years ago, years before present, years BP, you'll see. On my slides, that means years before present. I'll also use BP occasionally in a genetic perspective, which means base pairs, but hopefully you'll be aware of which one I'm talking about at the various times. So, so East Polynesia, so everything from central Polynesia, the Society Islands, that's Tahiti, and the Marquesas, up to Hawaii, Easter Island, and New Zealand, were really the last major push. But the western edge of the Polynesian Triangle in Samoa and Tonga actually was settled about almost 3,000 years ago, 2,900 BP are the earliest dates for there. And that's considered the homeland region then for the final push out into central East Polynesia that really began, and archaeologists, of course, are debating dates all the time, but the dates kind of now, I suppose some consensus is around 1200, possibly as early as 1500 BP for the first expansion into settlement into central East Polynesia, and then Rapa Nui and New Zealand settled about 750 years ago. Hawaii possibly a bit early, maybe as early as 1,000 years ago. But when we look at Pacific origins, all of the archaeological evidence, the linguistic evidence, and the genetic evidence tell us that these populations came from the West, from Southeast Asia and New Guinea. I say lots of debates about dates. This is kind of one of the more recent syntheses and what we've often referred to as chronometric hygiene, and really cleaning up the radiocarbon dates and really taking only those that are acceptable. And so this was published in 2011 and gives you kind of it was a Bayesian analysis of radiocarbon dates for the settlement of Polynesia. So Central East Polynesia wasn't settled really, they argue, before 1000 AD. Hawaii, Rapa Nui, and Aotearoa, New Zealand, 1280, or about 750 years ago. But Polynesian origins lie further back in the Western Pacific, and they're associated culturally, archaeologically, with what we call the Lapita Lapita culture and the Lapita cultural expansion. Lots of debates about the origin of Lapita. We don't actually see the full components of what we use to define the Lapita culture, the dentate stamped pottery and settlement patterns and so forth, until the Bismarck Archipelago there in this region here, about 3,350 years before present. And very rapidly, people moved through what we refer to as Near Oceania, an area that had been settled for tens of thousands of years ago. We've got people in the Solomon Islands by about 30,000 years ago, and people in Australia and New Guinea at least 50,000 years ago. Perhaps even now they're pushing those dates back to possibly even as early as 60,000 years ago. So in Near Oceania, we have one of the earliest out-of-Africa migrations with people in that region very, very early on. While Neanderthals are still running around in Europe, we've actually got modern Homo sapiens in Australia and New Guinea. And then Polynesia, and East Polynesia was the last landmass, that was the last region that was settled by humans. So it's a fascinating place to be studying human evolution and human history. Now, linguistically, all of these people in Polynesia and many of the people in Near Oceania speak languages that belong to the Austronesian language family. And so by combining the linguistics and the archaeology and the biology, we try to address the question of origins of the Lapita peoples. And lots of debates about the specifics. The languages trace back to Taiwan. The genetic markers don't really appear as full kind of Pacific lineages until someplace in kind of Sulawesi, Halmahera Island, southern island, Southeast Asia. And the archaeology, the actual Lapita culture doesn't appear fully formed until they reach the Bismarck Archipelago and the islands just north of New Guinea. So lots of interest. But all of this archaeological and biological and linguistic data, of course, challenges some of the earlier hypotheses that we've had about Polynesian origins possibly coming from the Americas, in the work of Thor Heyerdahl and his demonstration with the Kon-Tiki that actually you could sail a raft from the Americas to the Pacific. And this, at the time that they were doing this, and the biological data was still pretty sketchy, and they were just looking at blood group data, and it was, consistent. There were similarities. There were some genetic markers that linked Native American populations and Polynesian populations. We now know that that's probably from common origins in East Asia, and Native American populations went across the Bering Straits and down into the Americas, and those same kind of source populations then also moved southwards through island Southeast Asia and out into the Pacific. But we do have other evidence of contact between South America and Polynesia, in particular the sweet potato, or kumara as it's known in Maori, umara and various connotations of that elsewhere in Polynesia. And we know that this is a South American plant. It is not native to the Pacific region, but it appears in archaeological sites in the Pacific clearly before European presence. We actually have charred remains of sweet potato from a site in the Cook Islands in central Polynesia, dated to about 1,000 years ago. So, and again, archaeologists, lots of debates about the dates and how reliable those dates are, but they were clearly a major staple for East Polynesia in prehistory. The term kumara has been linked to the Quechuan word kumar, so it's not only the actual plant itself, but the name for that plant. So therefore, unless, as we always said, it was floated out, naturally on a raft with a little tag saying, "Call me kumara and plant me in the ground," we think it's pretty clear evidence that there was connections between the people, conversations between the people that allowed them to actually take that name for the item. We also have evidence with the bottle gourd, another American species that was transported and was important in Pacific populations. So just the presence of the kumara in itself and the archaeological evidence is evidence of contact. More recently, we've got genetic evidence of actually 3 introductions of sweet potato into the Pacific. The red arrows there, clearly the pre-European, and I would argue, and the authors argue, Polynesian dispersal. Polynesians making it to the Americas and bringing the sweet potato out, and it gets dispersed throughout the Polynesian Triangle. But then there's also the expansion from Mesoamerica by the Spaniards and taking the camote from Central America into the Philippines, and it gets dispersed through interaction spheres through island Southeast Asia. And then you have the Dutch also taking a variety from the Caribbean to the Dutch East Indies, and again, that sweet potato gets dispersed. So the genetic data has actually identified these three different lineages and different types of sweet potato that are being transported into the Pacific. So the genetic data are similarly, we've also got data about the bottle gourd indicating those connections. My foray into this field initially came about by looking at using the DNA of animals that people carried with them in their canoes when they crossed the Pacific as proxies for humans. We couldn't, at the time that I started doing my PhD research, study human genetic variation in the Pacific because of similar political issues to dealing with Native American DNA analyses and working with those communities and the way that those communities had concerns about the way biologists and biological anthropologists worked with them. And we're getting through a lot of those issues now, I'm pleased to say. So I realized that if we could figure out where the animals came from, Pacific peoples carried dogs, pigs, chickens, and rats with them in their canoes. They brought their important food items, animals and plants with them as they settled the Pacific. If we could figure out where those animals came from and what the genetic relationships were between those animals, it was a proxy for figuring out where the canoes came from and therefore where the people came from. So I started off in New Zealand. I started looking at the DNA of the Pacific rat, Rattus excellens, which is a distinct species from the European introduced rats and doesn't interbreed with those rats, which is very useful. And so I looked at the mitochondrial DNA of rats from New Zealand and compared those to rats throughout the Polynesian Triangle to determine where the New Zealand rats, or Kiore, were coming from. And we identified some interaction spheres within Polynesia and then extended that back into the rest of the Pacific. So we showed that the model worked. We could use DNA of these animals to trace the human movement. And so I moved on from the rats to look at archaeological samples and ancient DNA analyses of dog bones and pig bones in the Pacific, and was presenting that work at a conference in Chile when I met a man who became a very close colleague, who's become a very close colleague, who had told me that that he had identified some chicken bones in a pre-Columbian site in Chile, the site called El Arenal in south central Chile, just south of Santiago, right on the coast. And would I be willing to do a DNA analysis on the chicken bones? Now, at that point, I always knew from the Kumara data and so forth that, of course, Polynesians, the greatest voyagers in the world, who were on a trajectory moving across the Pacific, 1,000 years ago, why would they stop? They would have continued until they had to stop. And the only thing that would make them stop was hitting land and not being able to go any further. So I always was open to the idea of Polynesian contacts with the Americas, but I thought our chances of finding any actual evidence of that was going to be pretty hard, the needle in the haystack. We had the Kumara. To me, it was obvious. The greatest voyagers were the ones who went and got it and brought it back, and all kinds of interesting stories about why they selected the Kumara and not corn or some other plant that of South American origin. But when Jose Miguel Ramirez from Chile said that he had pre-Columbian chicken bones, I thought, interesting, okay. Chickens are supposed to be introduced to the Americas by Europeans, and you're sure that it's a pre-Columbian site? Yes, I'm absolutely sure it's a pre-Columbian site. I said, okay, sure, we'll have a look at it. And at that point, I just had a PhD student was starting her work, Alice Story, and she was looking at Pacific chicken bones and adding to that animal story. We had done rats and we had done dogs and we had done pigs, and so the last one we had to look at was chickens. And Alice was doing her PhD, so the next time I was heading in this direction, stopped off in Chile and picked up the chicken bone from Jose Miguel and brought it back to give to Alice to analyze. So she'd been looking at archaeological remains of chickens throughout the Pacific, and we found chickens in the earliest Lapita sites in near Oceania, at Musau, and Watum, another important lepidosite. And then we looked at the distribution of chickens in remote Oceania, and the part of the world where we were trying to address Polynesian settlements, and lots of interesting patterns and distributions. Chickens didn't go everywhere. The only animal that got everywhere that Pacific colonists went was the rat. The dog and the pig and the chicken have much more patchy distributions. For example, we didn't get the chicken. Chicken was not introduced to New Zealand, or successfully introduced to New Zealand, and we have to ask ourselves perhaps why. The pig didn't get there either. We only got the dog and the rat in New Zealand. Easter Island only got the chicken and the rat. They didn't get the dog or the pig. You know, they're again, I'm happy to discuss reasons for that with people if you're interested afterwards. So Alice, we gave her the chicken bone. We thought, okay, first we're going to get a radiocarbon date on it. And I was absolutely floored when the radiocarbon date came back at 2 standard deviations, clearly pre-Columbian. So you can see the dates there. The red arrow indicates 1492, which is, you know, Columbus sailed the ocean blue, and so it couldn't have happened a European introduction prior to that. And the dates at 2 standard deviations dated to 1307 to 1423 AD, so clearly pre-Columbian. So we said, okay, that's interesting. Let's do the DNA analysis on it, what we found was that the LRNL chicken bone gave us an identical sequence to the sequences that Alice was getting from 2,000-year-old bones from Samoa. We've got them from Vanuatu. All of these bones in the Pacific, identical DNA sequences. We had these results independently replicated in another lab. We gave them the bit of the bone. We had to split the bone in 3, so we sent 1 bit off for radiocarbon dating, and 2 other bits that we did one sequencing of one, and another lab in New Zealand sequenced the bone, and we got identical sequences. So we were pretty excited about that, and we wrote that up in PNAS, that we had radiocarbon and DNA evidence of pre-Columbian introduction of Polynesian chickens to Chile. So all very exciting, lots of press coverage, yay, yay, we finally have biological evidence, and it's academia, so of course there were serious critiques. To a certain extent, maybe, fair enough. We only had 1 bone, okay? We always want to get more than 1 radiocarbon date, of course, for samples, because we have anomalies in radiocarbon dating, and there can be those kinds of things. Some people suggested that perhaps people who are familiar with radiocarbon dating, apparently when an organism ingests marine foods, it can affect the chemical makeup of the bone that will make the radiocarbon date look older than it actually is. So they said maybe the marine diet, maybe it's not really as old as the radiocarbon dates suggest, and we're talking about a period pretty close to European contact. So that was questionable. And some people came back and said, perhaps the DNA, perhaps it's contamination with modern chicken DNA. Now, we had done all of the replications, independent replications, and everything that were required of ancient DNA, but okay, you know, the onus is on us to prove something extraordinary. So we got 2 more chicken bones from the site, had those radiocarbon dated. They were also pre-European, clearly. We also looked at the other dates based not on radiocarbon dating, but on thermoluminescent dating of pottery from the site, and all of the archaeological dates from bone, from pottery, were consistent throughout the site. There was no evidence of European artifacts or anything in the site. We thought we had pretty good evidence of pre-European chickens in the Americas. Now, was it Polynesians who brought them? Well, we have identical sequences in Polynesia, so we think we have pretty strong evidence for that. What about the marine dating? Well, Alice, bless her soul, she, as is a good PhD student does, takes up a whole new kind of area study and looks at all the isotope data for both the chicken bones and what isotope data we have available for animals from that region, eating the plants and the resources in the south coast of Chile. And what we found were that the chicken bones were eating a diet similar to the camelids, okay, llamas and alpacas and guanacos and things in South America. They were not eating a marine-based diet. It was a terrestrial diet, so therefore the dates don't need to have marine corrections, which would potentially make them older. We actually did the marine corrections even if just to check it, and they still didn't come out as post 1500. If anything, it was possible that they were eating maize, and maize does the opposite things to radiocarbon dates, and actually they could be older than the radiocarbon dates that we have, because maize is a C4 plant, which is, I'm not going to go into isotope analyses, but that's the way that they affect the dates. So we also sequenced those 2 additional chicken bones, and the sequences consistently came back not identical, but in the same haplogroup or the same lineage as the first chicken bone that we had. Another group did a whole bunch of modern DNA chicken analyses, and there's some dates, some sequences up there that they found the same group, the haplogroup E. So we found 2 different lineages of chickens in the Pacific, haplogroup E and haplogroup D. Now, we started looking at, okay, where do we see these different lineages? Haplogroup E lineages we see in very early sites in the Pacific. Haplogroup D lineages we see in later sites and in East Polynesia almost exclusively. So we have actually both of those lineages in South American material in pre-contact and in early contact period samples from historic sites. So we started looking at the distribution kind of worldwide of these, or in the Pacific and the Americas, of these different lineages, and we came to realize that really it looks like there was a chronological separation of these lineages. The earlier samples are haplogroup E, and the later samples are haplogroup D. And a number of our other studies of human DNA and dog DNA started suggesting that it was actually a late migration that may have resulted in the expansion, the final expansion out to East Polynesia. And we're seeing different lineages of other animal DNA lineages coming out in a later migration. So we also have one sample here in Micronesia at an island called Feis that actually had both lineages in the same archaeological site, and we don't see the later lineages, the D lineages, coming in until after 1,000 years ago. So we think that there's a second introduction. So that's the DNA, that's the chicken data. We've been trying to get some more chicken bones. There are not a lot of them in prehistoric sites, so we haven't been able to sequence any more. But we're always on the lookout. So what are the other evidence? Now, Terry and his colleagues published this very, very interesting paper about the evidence for sewn plank canoe technology in the Pacific. Now, Pacific people, when they build their canoes, they don't just make a dugout canoe like you tend to see, for example, in Northwest Coast Indians, but they actually build up the gunnels of the canoe by sewing wooden planks together and building those up. So this is what we refer to as sewn plank canoe technology. And it's seen throughout the Pacific in different forms, but only in 2 places in the Americas. Up here with the Chumash and Gabrielino, and down in south central Chile with Mapuche. Nowhere else do you have this technology. Now the linguistic evidence, Catherine Clark, a colleague of Terry's, who's a linguist at UC Berkeley, has actually found linguistic connections between the terms for sewing of the canoes and so forth between Chumash and Gabrielino with, I believe it's Samoan, but Polynesian languages, links in terms, again, the technology, yes, a boat could wash ashore and they might see how they use this technology, but the fact that they're using similar terms and similar terminology indicates that there was contact and discussion and exchange of ideas, not just seeing something and thinking about it. So here's the a picture of a tomolo from the California area. Here we have an Easter Island sewn plank canoe. These are not the big voyaging canoes, but these are later historic canoes. So picture this much larger with sails and so forth, but it's the same technology that Polynesians used. And here we have an early historic photo of a Chiloé island in southern Chile with their sewn plank canoes there as well. Now, this is South America here. This is Chiloé Island, where we've got the stone plank canoes. And basically all the way from about here, south of Concepcion, to Chiloé Island, they have this technology. Again, we're talking just there, and again, just here. Now, when we look at how people are actually likely to get to South America or the Americas from Polynesia, we actually look at the wind patterns, okay, and currents. From New Zealand, okay, down here, these are the roaring 40s. If you miss the Chatham Islands, which is a little island off of New Zealand, and there was a fair amount of transport between New Zealand and the Chatham Islands, the next thing you hit is South America. You get blown through the roaring 40s very, very quickly, and we have historic records of people actually leaving Tasmania and ending up on the south coast of Chile alive in open boats. So it's certainly possible. You can't, however, and similarly, if you're going from Rapa Nui, you also end up right on that south coast here of Chile. You can't get directly back, sailing in the same direction that you came. You actually have to head north, okay, with the Pacific currents, until you get to about the Gulf of Guayaquil, which is where the Kumara is sourced. From there, you can head back out into Central East Polynesia, and from there, the Kumara gets dispersed throughout the Polynesian Triangle. So it's an interesting location in terms of where do we see the evidence of the stone plank canoes. It's on these places where Polynesians are making contact, likely to make contact, and then, of course, we have evidence of the sweet potato being picked up from someplace up here. Sweet potato doesn't grow down here, being picked up someplace here and being brought out back into the Polynesian Triangle. Similarly, it's not as strong, the evidence, in terms of the winds and currents for North America, but it's a similar situation. Where are you going to end up? You're going to end up in the Channel Islands. Now, if you're coming from the Marquesas or from Hawaii, this is the direction you're getting the reverse wind and current patterns. Now, I was again at a conference. This is why it's important to go to conferences, talking about this work, and a young man, cultural anthropologist who was working on water rights with the Mapuche, came up to me and he said, I've been dating stone plank canoes in rivers, waterlogged sites in Chile. And the earliest dates that we're getting for these canoes, they just appear fully formed in this state at about 1300 AD, which is really almost identical to the dates that we're getting for the presence of the chicken bones in Chile. So he's got sites from throughout this southern part of the coast of Chile, and has radiocarbon dated these waterlogged, these wooden remains, and they're coming in at about 1300. So really, really interesting results there. So we've got the stone plank canoes. What about other archaeological evidence? These are signs of a chief, speaking chief of the Mapuche in southern Chile, a clava, a club, a stone club. And this is the typical, Mapuche are the largest indigenous group in Chile, and they extend further up the coast. This is the standard shape of the Mapuche clava. They say it looks like it's a sign of a speaking chief, and it looks like the beak of a parrot. And they say that that's the representation there is a speaking parrot. Right along the coast, from Chiloé up to about where we've been working on an island called Mocha, they have this shape. Similar shape. You can see it looks still like a bit of a bird shape. We've also, this is the Chatham Islands, just off of New Zealand, a waihaka, a stone club of a chief. Okay. Looks similar? Okay, we can often have things that look similar, fish hooks and all kinds of things, they're practical. We also have, these are the Maori New Zealand waihaka, or clubs, again, chiefs of the clubs, and in the lower section, the Mapuche clubs. Remarkably similar. My colleague Jose Miguel, the one who gave me the chicken bone, found this in our local museum, the Otago Museum, and the label on this artifact was a stick used for a game similar to hockey from the Austral Islands. The Austral Islands are in French Polynesia there, between Samoa, Tonga, and Easter Island. They're in the southern, let's see if I can get this. There we are. Moving too quickly. Okay. They are just here, the australs. And this is this hockey stick type thing. This is the Mapuche national game, traditional game of Palin. We don't have anything else like this in Polynesia. They don't play any game like hockey anywhere else in Polynesia, but in the Austral Islands. Again, interesting, coincidence, maybe. Here we have a funerary site in Chiloé Island in southern Chile. And they have these amazing wooden carvings representing spirits and so forth. We have remarkably similar, typical funerary carvings on marae throughout East Polynesia. These are from, I believe, Hawaii. But we also have them in Easter Island. More Mapuche carvings. Okay. In funerary sites, important ceremonial sites. Look at the shape of the face, the top knot on the head, the positioning of the arms. And we have New Zealand and Rapa Nui. Remarkable similarities. The attitudes about death and dying, very, very similar. Again, we can often, have similarities, 1 or 2 or 3, but when they start adding up, we start seeing an interesting pattern. There's another southern Chile artifact called a toki kura. This is a stone adze-shaped necklace. Now, in Maori, toki means adze. Kura is stone. We have an identical artifact with an identical name, toki ponamu, toki, that's a greenstone adze. Notice that the toki kura are all made out of serpentine or green stone. Okay, similarity in shape, similarity in form, similarity in meaning, similarity of the words. These are not typical Mapuche terms, toki kura, but it is a Polynesian term, a Maori term, and it's a greenstone adze that's worn as a necklace. Terry produced here's a short-handled adze for making the canoe planks, the stone plank canoes amongst the Chumash. Okay. Don't know what the term for them is. It would be interesting to find out. So given all of this evidence, we started saying we've got chickens, we've got all of this artifactual evidence, we've got linguistic evidence, we've got the plants. What about other Polynesian introductions? Would they have taken dogs? Would they have taken rats? Might they have taken pigs even? If we could find some more animal evidence, we're adding more evidence to the debate. So I went over and met with Jose Miguel, and we started going through museum collections and faunal collections to look for rat bones. We started looking for South American rats and looked at them. They're not Rattus excellens, or the Pacific rat. I said, what about dogs? There are no dogs native to southern Chile. They only have a native fox. And so we said, okay, we should be able to tell the difference DNA-wise between a dog and a fox. But the early European accounts describe people in their stone plank canoes with dogs. And we have early historic photos of these dogs with people in their canoes. And look at these dogs. They're little black and white, short-legged dogs. I was talking to the curator at the museum in Chiloé, in southern Chile, and was asking her about dog bones, and she said, we have a lot of canid bones, but nobody's gone through and looked at them. But one of the unusual things is the descriptions in the early Dutch accounts of the people wearing dogskin cloaks. Have you ever heard of anything like that? Well, in New Zealand, we have dogskin cloaks. Oh, sorry, there are no dogs in Rapa Nui, by the way. So the whole Rapa Nui connection, dogs couldn't have been introduced to Chile from Rapa Nui, but they could have been introduced from New Zealand, where dogskin cloaks are a very important ritual ornament. And this is a description, a drawing, a sketch of by one of Cook's, or a reproduction of a sketch of one of Cook's ethnologists, who described the Maori dog as being black and white, short-legged, and looked remarkably like the dogs that are in the early photographs with the stone plank canoes of the people, the Mapuche, in southern Chile. So we have not yet been able to find any ancient Pacific dog DNA. We've been looking, and again, we have these connections. You might have, okay, Rapa Nui may be the closest placed to South America, but in terms of speed of getting there from New Zealand in the Roaring 40s, you end up in Chile pretty quickly. And as we say, we know that people have survived this. So we don't necessarily have to assume that Polynesian contact only could have come from Rapa Nui. Okay? It could have come from people who are making voyages of exchanging stone resources and so forth throughout the Polynesian Triangle. We've got obsidian going several thousands of kilometers back and forth. So the distances that we're talking here are within the realms of typical or reasonable Polynesian voyaging distances. So we've been looking at the DNA of dogs. We now know in the Pacific we have 3 introductions of dogs, at least 3 into the Pacific region, 2 into Polynesia. And so we're starting to do some work on South American dogs, Chilean dogs. So 3 dogs. We started looking at some dogs from the mainland site that Jose Miguel had been excavating, a site called El Olivar. We looked at the DNA from this sample. Unfortunately, it turns out that it's actually, well, from our perspective, unfortunately, it's a Native American dog. It looks like dogs from Mexico. So not a Pacific dog, but we're continuing to look for dog remains. In order to do this, we decided, okay, where are we most likely going to find evidence of Polynesian introductions? A small boatload of people arriving on a mainland where people have been settled for a long period of time are not going to make a significant genetic impact. Okay? But people arriving on an island might make a significant genetic impact. We know that Polynesians and their ancestors, Lapita peoples, when they were traveling, would not, when they came to a place that actually had people settled on it already, like New Guinea, didn't settle on the mainland. They settled on the small offshore islands. Okay? Whenever that happened, they'd always go to a small offshore island. They could get away quickly. They're maritime people. Off they go. So let's look at small offshore islands. Not a lot of them along the coast of Chile, but one of them, Isla Mocha, looked particularly interesting. So there it is there. The yellow pin there is the El Arenal site where we found the chicken bones, just 100 kilometers north. Okay. So we said, let's look at Mocha. So this is a picture of the island. Unfortunately, in the 2009 earthquake, the tsunamis wiped out the major town, and it's got a few hundred people living on it. National park there in the middle. But there was another reason to be interested in this island. In 1903, a Chilean anthropologist, anatomist, recognized that the shapes of the skulls from burials on that island looked remarkably like Polynesian crania. Okay? Polynesians are a particularly robust population and have some unusual characteristics to the crania and postcrania that have been described as the Polynesian phenotype or the Polynesian physical characteristics by a number of anatomists, but Phil Houghton from our department early on described this Polynesian phenotype. So when you view the skull from behind, it looks like a pentagonal shape. You can see on the right there, Polynesian crania versus a European or Chilean, as it turns out, or Mapuche crania, much more rounded. Very robust, heavily muscled. The mandible, the shape of the jaw, is in particular the position of the ramus to the body of the mandible, much more vertical, and the body of the mandible has this kind of curvature, which results in what they call a rocker jaw, that you put the jaw on a tabletop and it will rock. We see it in some other populations. We see it in extremely high frequency in Polynesians. And postcranially, the head of the femur or the thigh bone, the shape of the one, it's very robust, and with a different angle of torsion between the head and the shaft of the femur, but also in the head of the femur has an oval fovea capitis, that little indentation in the head of the femur, versus a round one in non-Polynesians. So we said, well, that's pretty interesting. Went to the museum in Concepcion that had archaeological remains. We were getting the rat bones and the dog bones and looking for more chicken bones. Said, by any chance, do you have any human remains from Isla Mocha? And they said, yes, right there in that cupboard, right next to you. So I opened that cupboard, and to my surprise, I saw these amazing crania. Okay, pentagonal shaped, very robust, rocker jaw, oval fovea capitis. okay, I think we're going to have to stay here a little bit longer. So actually, as it turned out, we couldn't stay longer, but we came back to that museum and studied all of the crania that we had from Mocha. These were the other characteristics, what are called superstructures, occipital superstructures, that come from really intense muscle attachments on the back of the skull, between the neck and the occipital region, and also on the side. These are really unusual, but we see them at reasonable high frequency in Pacific populations. So I talked to one of my colleagues, a physical anthropologist, who'd been studying crania from the Pacific and looking at these characteristics, and he said, I sent him pictures of these skulls from Mocha. He said, these have got to be from Guam or the Marquesas or something. I said, no, they're South American. He said, I've never seen anything like this on any population outside of the Pacific. So in comparison, there we have on the left, okay, crania from Isla Mocha. On the right, from Tirua, just across the 30-kilometer water channel on the mainland of Chile. Very gracile population. Okay. Mapuche come up to about here on me. Okay. Here we have this massive, very robust individual. Similarly, Polynesian mandible, mainland Tirua mandible. You can see just the different size of it and the different shape. This is typically Polynesian. Postcrania, here's the femur of the Mocha femur versus a mainland femur. Look at the size of the knees. There's a reason that the All Blacks are such amazing rugby players, big, robust, massive musculature, and we see this in the material from Mocha. I did the analyses of the crania, measured them all, and ran it through a program that called CRANID that looks at comparing them to a known database. And we've had 6 different crania in the museum collections from Mocha. 3 of them came out here with Pacific and East Asian populations, and 3 of them with American populations. When we looked at the most similar populations to the measurements of the crania from Mocha, they came out as being similar to Guam and Micronesia, and secondly also to New Zealand Maori crania in terms of their shape and form. So again, just looking at where do we see Isla Mocha is just right here, right in that pathway in terms of where people are likely to make contact if they're coming from the southern part of Polynesia. So based on all this evidence, I wrote a grant application, as you do, to a New Zealand funding agency, our Blue Science funding called Marsden, and we received funding from the Marsden to go undertake excavations on Mocha. The skulls that we had from the museum, we didn't have good archaeological context for. So we decided we would look at settlement patterns, we'd source the stone and other artifacts from the sites, and undertake ancient DNA analysis of fauna and any human remains that we recovered. Now, when we're looking at what are we likely to find in terms of genetic evidence, okay, who are the most likely people who would be making contact with the Americas? We know that Polynesian voyagers were almost exclusively men. Women didn't travel in canoes on voyages of exploration or trade. So it's likely to be male voyagers, Polynesians, and evidence of admixture. The language of love is universal, marrying and engaging with local women. So what we really needed to do in order to see evidence of Polynesian contact in the Americas is to have evidence from the Y chromosome, of evidence of Polynesian males, or autosomal from across the genome, which at the time that we were starting this was very, very difficult, and this is now where DNA research is going, is to look at whole genome data and look at admixture between populations. So, as I say, the technology at that point was really next-generation sequencing technology that allows this kind of analysis of whole genomes of ancient samples was really just getting going, and so we started trying to get some of this information. So first of all, we went and excavated in 2010. This is my student Karen Gregg, who's working on dogs, and another of our Chilean colleagues, Andrea Silenbrand, who is a lithic expert, is Jose Miguel, and another colleague of mine from New Zealand, Richard Walters. So we had Polynesian and South American archaeologists with us, and we collected DNA samples under pure kind of controlling for contamination processes. We went back the following year, 2011, and found an amazing site right near where some of the skeletal material that we'd seen in Mocha had been found. And we found a beautiful midden site, 2 meters deep, lots of pottery, lots of animal bone, lots of shell, and so forth. The archaeological evidence from the ceramics indicated that we had early period Petren complex, as well as the later period, the El Vergel pottery as well. Lots of incredible artifacts from the site, including compound fish hooks, which are pretty typically Polynesian. A range of fish hooks, stone points, bone and silver artifacts. This is a little wooden whistle, which was very cool. We found a burial urn with a neonate in it, which we excavated for ancient DNA. And while we were doing the work, the landowner that we were working with found the site that some archaeologists, some Chilean archaeologists, had been excavating previously and had left open, which was unfortunate. But he took us to the site, and we had a look. And during the 2009 earthquake, part of the site had collapsed. And we went and looked at the site, and what we actually found, just right here, were 2 distal femur sticking out. I actually found a patella in the pit below, which is the kneecap. I went, oh, that's unusual. Looked up, and here were these knees sticking out of the bulk there. Looking at the size of them, I said, I think this is a male, and at that point banned any of my male archaeological colleagues from the site, because I said, we're not going to possibly have contamination with your Y chromosome when we excavate this. So my female archaeological colleagues and I excavated this burial, which was in danger of being destroyed. It was falling apart, and Chilean protocol is that you recover anything that's in danger. So we actually found another burial next to him that was fine, which we left. But what we found was this. An amazing, very robust male, 173 centimeters tall. That's my height. Okay, with a tokikura, with that greenstone adze on his shoulder. This was the first time a tokikura had been found in secure archaeological context. Here it is, not a tiny artifact. It was pretty impressive. My colleague radiocarbon dated some bone from the burial, and we got a date of about 430 BP. This is a picture of the tokikura. Beautiful, beautiful, 20 centimeters long. I mean, it's an amazing. It's made of local serpentine, and these are the similar tokikura from the museum on the mainland. This is a photo of the skull of this burial. Again, it's a very robust male with Polynesian characteristics. Now, unfortunately, because this was not our site, it was a site that was excavated by some other colleagues, Chilean colleagues, we did not have permission to actually do any DNA analysis on it. And unfortunately, the politics at the time, they could have given us permission, but they refused us permission to analyze this material. So until they agree, we can't actually do any DNA analysis on this individual. But we have been able to do some DNA analyses on some of the teeth and bones that we recovered from the site just a little bit further up the hill. And we've been able to look at both mitochondrial DNA, which is maternally inherited, passed down through the maternal line, and we're starting to get some Y chromosome data as well, so looking at the male line. Now, we would expect mitochondrially that they're likely to be South American in origin, but let's see what we got. So mitochondrially, Pacific populations have mitochondrial haplogroup B, but so do Native American populations in both North and South America, but we have not been able to find any haplogroup B in the Mocha material. We've got D1 and C mitochondrially. Not, pretty much what we would expect. We wouldn't expect Polynesian women to be leaving children there, but perhaps Polynesian men. So looking at the Y chromosomes from what we would expect, Polynesian Y chromosomes belong to lineage C, C2A, and O3A, and possibly K, where South American Y chromosomes belong to lineage Q and 3B. So given the Y chromosome data that we have right now, we have 2 individuals. The best data that we have, we've been able to confirm 2 of the mutations on the Y chromosome that unfortunately lead to both the Q and the O lineages. So we can't say at this point we haven't been able to do it. We're continually working on this material as techniques improve, and we're hoping to get some whole genome data from some of these samples. But in the meantime, some of you may have heard in 2014 there were 2 papers that came out in Current Biology. One looking at 2 ancient crania that were found in a museum collection in Brazil that belonged to a group called the Botocudo Indians. Morphologically, they looked different, and the DNA analysis that a team undertook showed that they had Polynesian DNA, both mitochondrial DNA and across the whole genome. They in fact showed that there was no admixture. They were pure Polynesians, okay? And they were dated to a period before Europeans were moving into the Pacific, but after a time that Europeans had contact in Brazil. So they argued that this was evidence of Polynesians in Brazil. You would think I'd be jumping up and down during the happy dance. No, we don't expect a pure Polynesian to be in Brazil, okay, at that point in time. So I was somewhat dubious about this, and I said, you know, working with museum collections, I've seen skulls kind of and material being moved around and getting misplaced and so forth. And we know that there was a whole trade, you know, early on of people picking things up and bringing them to museums and so forth. I said, my call is that these are probably misplaced. Interesting story. We have now found that there was actually a loan of 2 skulls from the Museum of New Zealand in the late 1800s to the museum in Brazil of a Maori and Chatham Island crania. We're in the process of trying to track this down now. Then there was a study of modern Rapa Nui that showed that modern Rapa Nui here have some European admixture, not surprising, but also some Native American admixture. And working out the dates based on DNA decay and so forth, they estimated that it was possible that this could be pre-European Native American admixture in Rapa Nui. So the possibility of having an admixed population, which is what I would argue you would have to really justify and argue that we have biological evidence of exchange, okay? We might have it, but it's modern data. 2 days ago, this paper came out in Current Biology, sequencing 5 individuals from Rapa Nui, whole genome sequences. Okay? 3 of them are pre-European, 2 of them are just post-European, all pure Polynesian. Did anybody see the kind of news stuff on this online? Did anybody? Okay. The press got hold of this. No evidence of Polynesian contact with the Americas because there was no Native American DNA in Rapa Nui. And the mystery is solved. Rapa Nui are Polynesian. It's like, yeah. Now, we've all known that Rapa Nui are Polynesian, and because there's no Native American DNA in Rapa Nui doesn't mean that there was contact. There wasn't contact with the Americas. Again, that meant that they didn't bring people back to Rapa Nui. So I think the jury is still out. Again, you know, I'm absolutely positive that there was contact, but we've got to actually have reliable data to before I start doing the happy dance. So conclusions, we do have strong evidence for Polynesian contacts with South America. We have the sweet potato, the bottle gourd, chickens, stone plank canoes, the linguistic data, the tokikura, dogs, human morphological data on mocha, all of this stuff really, I think, is more than just a smoking gun. We need human genetic data. I think the data that we have is not conclusive. I'm not ready to say that that's the result. We need evidence of genetic admixture, of well-dated, pre-Columbian, in archaeological context, which is absolutely essential, remains either in the Americas or in Polynesia. So we are looking for that. Where do we look? Well, the islands. I think we have to look at the islands, whether that's in South America or Mocha, or whether potentially up here, offshore islands, and not necessarily Rapa Nui. Everybody assumes that it's got to be the closest island. These are people who are voyagers. They're moving all the time at speeds like you wouldn't believe. So we can't just say it's going to be Rapa Nui, and we have to look at other places as well and keep an open mind about it. So I would just like to, at this point, thank you all for coming, thank Terry, acknowledge my colleague Jose Miguel Ramirez, who really got me engaged with this question, my colleagues in Chile who helped us excavate the site, and my funding agencies. And of course, the blatant plug, if you're interested in this stuff, Terry and I co-edited a book on Polynesian contact in the Americas. You can buy it, unfortunately only in hardcover, I think, but yeah, and I'm happy to take questions.